Okay, our next human body system that we are going to be covering is the nervous system. The nervous system is covered in chapter 14 of our book, and we're going to be doing sections 1 through 4. And we're going to answer the following questions. What are the functions of the nervous system? What are the parts of the central, limbic, and peripheral nervous systems? Can you describe the anatomy of a neuron? How does a neuron send a nerve signal? So a little bit of physiology there. And then what functions do the central, limbic, and peripheral nervous systems coordinate? Overall, though, the functions of the nervous system are to sense internal and external changes, and then to allow us to be able to respond to those changes. If we recall back to week 1, we'll hopefully remember that one of the characteristics of something that is alive is being able to sense changes in their environment and be able to respond to that. So that's kind of the job of our nervous system. One of the second things that our nervous system can do, though, is that can also sense changes from the inside of our body. So what is the pH of our blood? We've talked a lot about how that's maintained and how important that is. But who is telling the kidney to take out more hydrogen ions? Well, that is a function of the nervous system. So basically being able to regulate and tell the other body systems when it's time to perform their most important functions. And then now we're going to integrate and coordinate all of the body's activities. So I'm simultaneously speaking to you, maybe thinking about how I could use an extra cup of coffee, and I'm moving my hands around in coordinated movement, even though you can't see me. But all of those things are being regulated and integrated together by my nervous system. And the nervous system is made up of nervous tissue. So let's do a quick review of nervous tissue. We hopefully remember that there are 2 types of cells that make up nervous tissue. We have our neurons and our neuroglia. And the basic unit of the nervous system is really the neuron. The middle part of the neuron, which is the cell body containing the nucleus and all of those internal organelles. And then coming off of the cell body, we have these like little root-like structures called the dendrites. And those dendrites are what's going to anchor the neuron into the surrounding tissue, which is the neuroglia. Coming off of the cell body in the other direction, we have our axon, which is going to end in our synapses or our axon terminals. The axon itself is surrounded in a fatty layer called the myelin sheath. My little H is missing there, myelin sheath. But that high fat, it insulates the axon. So when we look at an electrochemical impulse here in just a little bit, it helps that neuron send its message faster. And so when we're talking about nutrition for infants, and we're making it really, putting a high emphasis on the high fat content of food for infants, like high fat milk, that's because that is when the body is developing all of these neurons and developing that myelin sheath layer. And we want to make sure that those are really, really healthy because that's going to keep that neuron going longer. Because just like we saw with cartilage, neurons also have a very low capacity to regenerate or repair themselves once they're lost. So there are 3 main types of neurons. Their structure is all similar, although their shapes are different. Everybody's going to have a cell body. Everybody's going to have an axon. Everybody's going to have synapses or those axon terminals. The first type of a neuron is called a sensory neuron, and these transmit signals toward the nervous system, so basically towards the brain. They gather information from the environment, sense information from the environment, and then send the message up to the brain. So if you are sitting at your desk right now like I am, and you're fiddling with a piece of paper and you get a paper cut, that pain that you register was a message sent from the paper cut by a sensory neuron up to the brain, to the central nervous system. Secondly, we have motor neurons, or interneurons, they're sometimes called, motor neurons, they carry information away from the brain. So, for example, if our paper cut was the pain sense was sent to the brain from through the sensory neurons, and then our reaction, or what the brain tells our finger to do as a response to that pain, to hopefully be pulled away from the source of being cut, is going to be sent through a motor neuron from the central nervous system, from the brain, back out to whatever the signal was. And then our third type in the middle there are our interneurons, or relay neurons. And these are located between sensory and motor neurons, mostly within the brain and spinal cord. And these are the ones that are integrating all of the information received and sent out together. So 99% of all the neurons that we have are actually interneurons, and that's what allows us to speed up our processing, because they can kind of speak both languages and talk back and forth between them and integrate all that information together. No matter what type of neuron they are, the mechanism of sending a nerve signal is the same. So neurons are able to send messages to one another and to the cells of other tissues by conducting what we call this nerve signal. And nerve signals are electrochemical changes or impulses that are sent from cell to cell. But in order to do that, each neuron has to have some kind of energy to start with. It has to have a built-up energy source. Similarly to how you can't turn on a flashlight if your batteries aren't charged, you can't fire a signal from a neuron if there's not some initial source of energy. So each neuron on the body, when it's not conducting a nerve impulse, and it has energy stored up in it, so it's ready immediately when it's needed. And the energy stored in a resting neuron is called its resting potential. It's potential energy because it hasn't been released yet. Resting potential energy is created because the neuron maintains an unequal balance of charged ions on each side of its plasma membrane. So this is, again, really similar to how a battery works. We've talked a lot about ions moving naturally with the concentration gradient, but a neuron will actually use active transport. So we'll use ATP to pump charged ions against the concentration gradient so that there are more ions on the outside of the cell, outside the neuron, than on the inside of the cell. And this pump is called the sodium-potassium pump. The pump moves sodium ions out of the cell and potassium ions into the cell. And this movement of ions to a state of disequilibrium is what charges the battery or primes the pump for the nerve to be able to send a nerve signal. In order for it to be fired, a stimulus has to basically release that energy or activate the potential energy stored in the resting neuron. And once the stimulus has occurred, that's an all or nothing reaction. If the neuron fires its signal, it will fire it all the way and use up all of that stored energy. So if the stimulus is something that causes pain, for example, we only feel more pain when more neurons are fired. Not because a single neuron told the body that that hurt more, it's just because it affected more neurons. And when the nerve signal is fired, it must travel from the cell body down the axon towards the synapses. So these nerve signals are only sent in one direction, from the cell body down the axon towards the synapses, and it's the synapse that's going to be able, or the terminal, is also going to be able to send that message on to the next neuron. And this is done by using the stored resting energy to create an action potential. So through processes of repeated depolarization and repolarization of the axon. So let's look at that in just a second. So the stimulus occurred, which released our action potential, our resting potential. And now we're going to send that message down the axon using depolarization and repolarization. So this process of the nerve signal being moved along the length of the neuron to send its message to the next cell is called its action potential. The electrochemical charge is moved from the cell body down the axon towards the synapses through this process, depolarization and repolarization. When an action potential is triggered, protein channels that move sodium allow sodium ions to rush back into the cell. So before, we were pumping them all into the outside. Now we're opening up those channels and allowing them to come back into the nerve cell. This is called depolarization because the charge inside the cell changes from being negative compared to the outside to now being positive compared to the outside because of the addition of all of those positive sodium ions. Immediately after depolarization occurs, so once that change in charge happens, the channels that move sodium close and channels that move potassium open, causing potassium, another positively charged ion, to move out of the cell. Because the positive potassium ions leave, this makes the inside of the cell negative again compared to the outside. We call this repolarization because the conditions on either side of the cell are now polar again. After the signal has been fired, the potassium channels open again, allowing them to be returned to the inside of the cell, and the sodium channels move sodium to the outside of the cell, which restores the resting potential of the neuron while it then waits for another stimulus so that it can be fired again. I'm going to repeat this one more time because it's so important, but action potentials only move in one direction, from the cell body down the axon towards the synapses. Once it reaches the synapse, the nerve signal then has to be transferred from the synapse of that neuron that just fired to the next one, to the next neuron, so that it can go either back up to the brain or back out to tell the body what to do. And that process of sending the message from one neuron to the next is performed by neurotransmitters. Neurotransmitters are molecules that are stored in vesicles located in the synapses, or the terminals, the endpoints, and the action potential arriving at the synapse so that it did repolarization, depolarization, repolarization, depolarization, all the way down the axon, and now that change in charge, that electrochemical impulse, has reached the terminal, reached the end, which causes the neurotransmitter to be released into the synaptic cleft. And the synaptic cleft is the space between the two neurons. So here's the synapse of the first neuron that was fired. Here's the neurotransmitters waiting to be released. It receives the signal down the axon, says, tell the next neuron what's going on. The neurotransmitters are released into this space in between. The next neuron is going to receive the neurotransmitters via a protein receptor. Depending on the type of neurotransmitter that was sent, it can either excite or set off the next neuron, or it can inhibit it, which would stop the transmission of the nerve impulse. Because neurotransmitters have that ability to basically turn neurons on, they also need to be gotten rid of pretty quickly. So if the transmitter protein sticks around too long, then it will prevent that neuron from being able to go back to reaching that resting potential. I have a video that I'm going to post again in that the supplemental portion. So again, if you are a more visual learner and you want to see those ions moving around, in which direction they're going and how they're creating those differences in charge, that video will help you. I will see you in part 2 to wrap up our discussion of the nervous system.